So, finishing up meiosis with video 9. So this is a picture from the last video. Mitosis, meiosis 1 and meiosis 2. Mitosis, 1 diploid cell becomes 2 genetically diploid cells. They're all genetically identical, and this is a cycle. This takes place in somatic cells. It is asexual reproduction. Meiosis 1 and 2, this reaction will only take place in the gonads, testes, and ovaries. And this is the formation of the gametes. This is the formation of egg and sperm. Any other type of cell division is going to be mitosis. So meiosis goes to 2 PMATs. Meiosis 1 is 1 PMAT, meiosis 2 is the other. So let's just go through it one more time. Meiosis 1 looks pretty much like mitosis, especially with our microscopes. Goes to G1S, where the DNA doubles, monads become dyads. Everything else is going to double because at the end we're going to have 2 cells. But after that, there's going to be some things that are going to be different. Remember, the main purposes of meiosis 1, mix up the DNA. That's going to happen in prophase 1. The homologous pairs are going to pair up. Bivalents are going to form. The bivalents are going to cross over. They're going to grab each other. They're going to break DNA. Whenever they break, they're going to exchange DNA. So now every single chromosome, every dyad, is going to be a mixture of the mom and the dad's DNA. Now the difference we see is in metaphase 1. Another way to mix up the DNA is random alignment, where it's random of how they line up in the middle. And this is the only place you're going to have bivalents. So let's compare. Mitosis, single file. Metaphase 1, pairs. Metaphase 2, single file. Alright, let's continue on with anaphase 1. This is where the diploids become haploids. So notice these are dyads. Pairs go apart. After this, everything is going to be haploid. Let's compare it to anaphase of mitosis. Right here, the sister chromatids go apart. The dyads become monads because that's going to happen up here in metaphase. The centromeres have to double. You also see that over here in anaphase 2. Remember, the whole point of meiosis 2 is to convert those haploid dyads into haploid monads. Because dyads are becoming monads, in metaphase prior to that, centromeres have to double. So right here, let's go back to meiosis 1. Anaphase 1, after this, you'll only have one of every kind. Mitosis, we have 2 blues, we have 2 greens, we have 2 big ones and 2 little ones. Here, notice the big ones are going to separate. The little ones are going to separate. So after this, there's going to be 1 big one in each and 1 little one. When we get to telophase 1, they're going to be genetically different. They're going to be dyads. Let's compare it with telophase of mitosis. Notice monads, and they are diploids. And these are going to be genetically identical. Over here, telophase 1, they are dyads, genetically different from each other, and they're haploids. Telophase 2, they are haploids, but they are monads. Meiosis is not a cycle, so it's done right there. So this is a good picture to study. And another difference is comparing meiosis 1 and meiosis 2, right here, interkinesis. So a big difference between mitosis and meiosis 2 is interkinesis. So the whole point of meiosis 2 is to convert those haploid dyads into haploid monads. So we don't want to double the DNA. But we do, notice 4 cells, we do want to double everything else. So interkinesis is like interphase. Proteins double, organelles double, but it skips the S phase and the DNA does not double. That would ruin the whole purpose of meiosis 2. So at the end, 4 genetically different cells, monads, and it is not a cycle. They're finished. Here's another picture we can look at. Meiosis. Meiosis 1, interphase. Prophase 1 is trying to show you they're crossing over. Diploid number is 4. Look, we have 2 red ones and 2 blue ones. The green is blue. This is a crossed over exchange DNA. Anaphase 1. Notice the pairs are in the middle. Anaphase 1. Notice those are dyads that are going apart. Telophase 1. Telophase is the cytokinesis actually beginning in anaphase 1 that it's going to complete in the telophase and beginning of prophase 2. So over here we had 4 cells, dyads, now we have 2. So all of this is haploid. Because now we have one of each kind. We have one of these greenish colors and one of these reddish colors in each of these. So notice we're having 2 cells with a haploid number, and both of these are going to go through the same process. Metaphase 2, they line up in the middle. Centromeres are going to double, so look over here, anaphase 2, it is the sister chromatids go apart. Anaphase 1, look, they were dyads. Cytokinesis is actually going to begin in anaphase, starting a little deeper, then complete. At the end, you're going to have 4 cells. Mitosis, move it up a little bit. Goes through PNAT, ends up with 2 cells that are diploid. We can make a chart like this. If you make charts like this, it will be really, really helpful to study. DNA synthesis, process, S phase, and meiosis, also in the S phase, interphase, and mitosis. Synapsis of homologous chromosomes occurs in prophase 1 of meiosis. Does not occur in mitosis. Crossing over. Prophase 1. Does not happen in mitosis. Alright, mitosis is asexual. That means there's no mixing up of the DNA. Process. Homologous chromosomes are the pairs of bivalents. Line up in the middle, the equator, metaphase plate, all the same thing. Metaphase 1. That does not happen in mitosis. Sister chromatids line up at the metaphase plate. Alright, so dyads line up single file at the equator. That does happen in metaphase 2, and it does happen in metaphase of mitosis. Remember, genetic composition of the daughter cells. Meiosis ends up, right at the end of meiosis 2, 4 haploid cells, genetically identical, monad chromosomes. Mitosis ends up with 2 diploid cells, genetically identical, monad chromosomes. So here's meiosis in a plant cell. So dyads line up in the middle right here. Metaphase, right? Dyads going apart. Cell plate is starting to form. Becomes 2 cells. Notice they're half of the size of the original. Alright, so they're in the middle, so I would probably call this one metaphase 1. Going apart, dyads are going apart. This will be part of anaphase, telophase, cell plate. And since the dyads have separated, these are going to be haploid. Alright, so that was in meiosis 1. Let's go to meiosis 2. Here, they're going to be lining up single file, no longer pairs. Alright, now notice right here, there's the sister chromatids that are going to go apart. Let's go like 2. You know, look at this. This is a really interesting part. This is going to be an easy way to recognize it. Remember the plant cells at first, they're going to be smaller, and they never completely separate. Remember those plasmodesmatas, those connections? From lab in our lecture. So look right here. 4 cells. So if you had a test, you had this one right in here and this one right next to it, you would know that this is telophase 2. It has 4 cells, especially when it's next to a bigger one. And you know it has to be meiosis, not mitosis, because there's 4 cells. Having gone through meiosis, we're going to see it's not quite so straightforward. Meiosis is called gametogenesis, formation of the gametes. Genesis. It is actually not going to be the same in formation of sperm as the formation of eggs. Spermatogenesis, you do not need to know all these details for the test. Just know, in spermatogenesis, taking place in the testes, we have 1 cell that is actually going to become 4. So they're going to, for every one, they're going to form 4. And half of them are going to be X's. And half of them are going to be Y chromosomes. So there's going to be the male who determines the gender of the offspring. So formation of the sperm. Genesis. So it's not pronounced oogenesis, it's pronounced oogenesis. Formation of the eggs. Ovum. And you do not need to know these names, but here's something that you need to know about. In the female, just think about a chicken. There's a big egg, right? So again, think about the chicken. That chicken gets all its nutrients from what is inside the egg. So what we have here is we give it a different color so everything's not the same. For oogenesis, we have unequal division unequal division of the cytoplasm. So that, like a chicken egg, they'll get a lot of the nutrients in one. So they actually will only be able to produce unequal division. Hold on, just pay attention to what I'm doing. Of cytoplasm. So in this case, there actually is only going to be one egg. Unequal division of the cytoplasm will cause a polar body. Let me make that a different color. Make it purple. So you have unequal division of the cytoplasm. So that one is going to receive the majority of the cytoplasm, so all the nutrients. Okay, so in Meiosis 1, unequal division of the cytoplasm. And these are just going to be polar bodies as well. None of these can actually be fertilized. Here again, another polar body. Now sometimes these polar bodies actually remain attached to the eggs, and that's actually a benefit for the offspring. That means extra nutrients. Okay, so spermatogenesis taking place in the testes produces 4. 2 X's, 2 Y's. We got little tails here. So these are motile. These are ones going to be moving. Motile. They can move. Oogenesis in the female has unequal division of the cytoplasm. Think about the chicken growing inside the egg. Okay, all the nutrients for that little baby chicken when it's growing only comes from that egg. Alright, so one egg. These polar bodies cannot be fertilized. Sometimes they do remain attached to the egg. And if they do, extra nutrients for, in this case, the chicken. Think about seeds. A corn seed. So corn seed has unequal division of the cytoplasm. A lot of the nutrients are going to go into that seed. Because that plant, at first, is going to get all its nutrients from what is inside the seed. Same thing as this. So let's look at some other differences between spermatogenesis and oogenesis. So here's another difference here. As spermatogenesis actually does not begin, it's not activated until puberty. Sperm are going to last longer than the eggs, and the male, that's a symbol for male, can, depending on the health of the male, they can produce sperm 80, whatever. So again, they're going to be producing 4. Half our X's, half our Y chromosomes. Oogenesis, formation of the egg, has some major differences. One egg only is an unequal distribution of the cytoplasm. Now polar bodies. So when the oocytes, the egg cells split, one is going to be a lot larger than the other. Produce one egg, 2 to 3 polar bodies. So that means that's a symbol for female. Asymmetrical cell division. Now here's another big difference. Females are born with all the eggs they're ever going to make. They don't make any new ones after they're born. So, girls, the ovaries contain over 6 million eggs. This decreases to about 1 million by birth. And by the time they reach puberty, it's going to be reduced to be about 400. So they produce all the eggs they're ever going to have before they're born. And they're actually going to be in the prophase 1, the chiasmatic stage, or the crossing over stage. So they're present at birth. They're not going to be released until puberty, 1 per month. And we'll talk about that in just a little bit when we talk about something else very different about them. Something called meiotic arrests. But before we get to the meiotic arrest, there's some other differences. Then we talk about there's more sperm than eggs, 4-to-1 ratio. Male process continues longer. Eggs are larger than sperm, and sperm are motile, but the eggs are not. They sit and wait to receive the sperm to get fertilized. Alright, now let's go look at the meiotic arrest. Only the females in oogenesis have meiotic arrests. So again, meiotic arrests are only in oogenesis. So I told you that the females are born with all the eggs that they're ever going to have. And they form them when they're inside their mother. The eggs are not mature. They are in the stage of prophase 1. In the chiasmatic stage and crossing over. And they stay in that stage until the girl reaches puberty. Then, a particular hormone, the follicle stimulating hormone, causes one of those eggs to be activated per month. And only then will that oocyte continue on with meiosis. So it will continue meiosis 1 and continue on to meiosis 2 until it gets to metaphase 2. First arrest is in prophase 1. When they're inside their mom. Second meiotic arrest is metaphase 2. And it will stay in metaphase 2 until it's fertilized. If it's not fertilized, then it will disintegrate. But only when it actually is fertilized will it complete meiosis 2. So unequal division of the cytoplasm. So again, we're only talking about one of these. Only one is an egg. We're talking about that. So FSH, the hormone on here, that causes one of those eggs to be activated, to be released during puberty. That's why it's called this. That's why it's called a period or menstruation. Period is something that's happening over and over and over again. And if you have fraternal twins, instead of having 1 per month, you could have 2 per month, and if both of them are fertilized, right, it can just result in 2 children. So this is only in oogenesis. So gametogenesis is the formation of gametes, or sex cells in animals. Gametes are sperm and egg in humans. Fertilization occurs when sperm and egg nuclei fuse. And let's go look down here. Gametogenesis occurs in the testes, make sure where it takes place in males, where spermatogenesis produces sperm. Gametogenesis occurs in the ovaries of females, where oogenesis produces oocytes, which are called eggs. Gametogenesis involves meiosis, a process that reduces the chromosome number from a diploid to a haploid number. In sexually reproducing species, if meiosis did not occur, the chromosome number would double with each generation. Meiosis consists of 2 divisions of 2 PMATs. The first meiotic division, meiosis 1, and the second meiotic division, meiosis 2. Therefore, you would expect 4 haploid cells at the end of the process. Indeed, there are 4 sperm as a result of spermatogenesis, In females, meiosis 1 results in a secondary oocyte and one polar body. The polar body is a non-functioning cell that will disintegrate or remain attached. Some nutrients. A secondary oocyte does not undergo meiosis 2 unless fertilization, fusion of egg and sperm, occurs. At the completion of oogenesis, there is a single egg and at least 2 polar bodies. Again, there could be 3. So we're going to look at this picture here, this figure, which is going to illustrate that. So spermatogenesis produces 4 viable sperm. Oogenesis produces 1 egg, 2 to 3 polar bodies. So the secondary oocyte does not undergo meiosis 2 unless fertilization occurs. In humans, both sperm and egg have 23 chromosomes, so they're haploid. Therefore, following fertilization, the zygote has 46 chromosomes. So let's look at the picture. Alright, let's look at the top picture first. Spermatogenesis taking place in the testes. First female, first meiotic division. 2 dyad haploid cells, genetically different from each other. That one's an X, that's a Y. Goes through meiosis 2, ends up with 2 X's and 2 Y's. So 4 monad haploid cells. Every single one genetically different from each other because of prophase 1 and random alignment in our first meiotic division. Alright, I just hit the invert button so maybe you can see it better with this. So the second picture. Alright, we have meiotic arrests. We have 1 in pre-birth, prophase 1. So when they're born, females are born. It's the crossing over stage of prophase 1. Stays in that stage until puberty, follicle stimulating hormone, causes ovulation to happen. There we go right here. That's going to go on, finish our first meiotic arrest, and continue on until it gets to metaphase 2. And it's going to stay in metaphase 2. Alright, metaphase 2. If it does not get fertilized, it will disintegrate. It's called menstruation, during a period. But if one of these sperm come and fertilize it, it will actually continue and complete meiosis 2, finish phase 2. So here's some polar bodies that can remain attached as extra nutrients. And once they get together, then we have mitosis taking place. So after that, we're going to be having mitosis take place. Alright? Oops. Mitosis. It's amazing when these two get together, it's like a big spark of light. They're actually taking videos of it. So I mentioned the other day that one man, one woman could have, if they could have, a billion children. None of them would be exactly the same. We're not talking about twins and everything like that. So let's look at this. Due to independent assortment, random alignment, independent alignment, chromosomes are distributed to daughter cells in various combinations. The total number of possible combinations is 2 to the n, where n is going to equal the haploid number of the chromosomes. In humans, our haploid number is 23. So look at this. The number of possible chromosome combinations just due to independent assortment random alignment is this. Pretty big number. More than 8 million. Alright, but we know there's two ways to get variation. Crossing over. Let's just say there's only one. If there's just going to be more than one, let's just say there's only one. The total number of possible combinations is 4 to the n. In humans, independent assortment, alignment, and one crossover, so we add one crossover. So against this one, usually there's going to be more. It will result in 4 to the 23rd different possible kinds of gametes. Look at this number, just one crossover. I'm not even going to read it. So we have random alignment crossing over. And then, due to fertilization, switching all three of these, egg and sperm are combined in humans. This will result in 2 to the 23rd to the second power. Oh, look at this. This is just with random alignment. Here's how many different possible offspring are possible, just with random alignment, not crossover. Now we're going to add one crossover. Alright? For the 23rd to the second. I'm not using it. I have no idea what that number is. That huge number of different offspring are possible per couple. So, meiosis is amazing. It's amazing at mixing up the DNA through random alignment, crossing over, and then fertilization. Make sure you know all three of those. So what is the value of sexual reproduction? The key thing is that you get genetic variation. Genetic recombination in metaphase 1, independent assortment of chromosomes, random alignment of homologous chromosomes, crossing over, prophase 1, mix up the alleles across homologous chromosomes, random fertilization, which sperm is going to fertilize which egg. Why is that important? Because it decreases your chance of going extinct. Provides variation for natural selection. If everything was the same, like remember the potato famine in Ireland that you looked at in the video? They didn't have very much variation. The disease came, fungus, and wiped them out. So the value of sexual reproduction and the value of meiosis is to get genetic variation, decreases your chances of going extinct, increases your chances of recovering from illnesses, etc. Alright, well, that is the end of the information for test 4. We finished meiosis and mitosis.